Hello? Yo, yo. Okay, it's very. Yeah, okay. I'm just testing a bit. Oh, well, let me check this. You can probably hear me, but. Uh, what about this? Hello, 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 hello. Testing, testing. What's up, brother? Okay, that was a bit better, but yeah, well, whatever, man. This OBS shit is not my gig. I usually stream at Streamlabs, but yeah, I think it's time to. Oops. I'm just gonna do something with the settings a bit. Uh, mm, 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 mm. Yeah, you can probably hear Breton now. Probably. I don't know. Alright. This is just a test. Uh. Yeah, I don't know if the desktop audio works and the shit, bro. It's. Alright, okay, it works. It works pretty well. Yo, what's up? Welcome back to another stream. Today is Wednesday, I think. Wednesday, yep. And yeah, so tonight, we are going to do another study stream and another, well, making my video done about orbitals, geometry, all that, like we did last stream, and also uploaded my first short. Well, my second short now, but it's going pretty well. Currently, let me check. Okay, I got. Can you see this? Okay, I got 36 on my first short with 1 like, and I got 6 view and 0 likes on my second. So that's pretty great progress, honestly. But yeah, hope you're all doing well. And let's just get this shit going, honestly. And yeah. But yeah, for me, honestly. Oh, wait. Can you even hear me? Probably can. Why did they mute? Okay, everything works apparently. So I'm just gonna roll with that. Alright. So, let me see here. There we go. We got the transition. Oh, smooth as shit, brother. Anyway, alright. So, like I said in my yesterday stream, orbitals are where electrons live. So like their houses basically, where they like occupy in the region, or like the probability where you can find an electron. And electrons decide where bonds can form, so they can decide whether a sigma bond or a pi bond can form, or a triple pi bond. So a sigma bond is like this one line. This is a sigma bond. Okay, that was a really ugly ass sigma. This is sigma bond, and this is a pi bond, which is double bond. And this is just a triple bond, or also known as a pi bond. So yeah, it's all about electrons, man. When coming to chemistry, it's all about electrons, like how they move and where they're gonna move and all that. So it's pretty easy. Yeah, so they also decide what angles those bonds can form. Like, some can form like tetrahedral shape, or triangular planar shapes, or liner shapes. Like, if you're talking about tetrahedral, methane, CH4 looks like this basically. So we have there we go. Probably like this. And then this one goes out, and this one is, well, out of the plane. There we go. Like this. This is a tetrahedral shape, and when you see dashed lines, like this one, a dashed line, that means it's going outward the paper, like away from you. Like if you're having bad breath, then the H is going away from you. When you see the wedge lines, in the wedge, dashed lines, and when you see a wedge like this one, that means it goes in front of you, like it wants you, just like your crush it wants you. So yeah, this is a tetrahedral shape. Yeah, don't yeah. So this is what a tetrahedral shape looks like, and then we have a triangular planar. Let's say this compound, just B, and this is triangular planar, and this is liner. This is also carbon dioxide, also known as CO2. So yeah, and the whole of this, basically this is a thing called Vesper. So I will definitely make a video for that, like going in depth for that, because it's very useful, and it's mostly in inorganic chemistry. So in organic chemistry we only have like three, so tetrahedral, trigonal planar, and liner, but there's also some more. Yeah. So we have this, how orbital forms. A chemical bond forms when atomic orbital. I can't even speak. Chemical bond forms when atomic orbitals overlap. So, like I said, we have this tetrahedral carbon, and it has 4 orbitals. Orbitals is like electrons' houses where they occupy in, and then hydrogen atom with the s orbital, just a circle round, has appeared. We add 4 H atoms, and then they overlap. They overlap and share electrons. And oh shit! And this this right here. This is what happening. It's like they're kissing, basically. So when they overlap like this, like this, those yellow ones are electrons. This right here is a covalent bond. Can you even see? Wait, this is a covalent bond right here. So yeah, then it's a p orbital and a s orbital hugging. Yeah. Let me check if my audio still works, because I'm a bit skeptical. Yeah, it works pretty fine. So, then we have, whoa. There we go. Then we have, yeah, the only orbital we only care about in organic chemistry is s orbital and p orbital. I should probably make my thing more like smaller. There we go. This should be much better for the audience. Yeah. And yeah. So atoms in the second row of the periodic table comes with all basic setup. So CNOF and yeah, 1 s orbital and 3 p orbital in the valence shell. They combine then and form hybrid orbitals, which is sp3, sp2, and sp, which is called hybridization. Then I need to like fill out what the hell does that even mean for each of them, so it's easier for the viewer to like understand it very easy. Because in the exam there's gonna be like a question like what hybridization does this carbon atom have? Then you have to be like woke and know what the hell is going on. Yeah, why hybridization matters. So each hybrid orbital can do one of 2 things. They can form a bond with another bond or hold a lone pair of electrons. So orbitals can either form a bond, either a sigma bond or a pi bond or a pi bond, or they can hold a pair of lone pairs of electrons. And how to determine hybridization? The way easy way. Okay, so we have to find the hybridization of an atom. We have to count how many atoms it's bonded to, basically. So, like if we have if we have acetone, like a C and then a double bond and then a O and then a CH, and then we have, I can just draw it instead of explaining. If we have like this, as its own, also known as nail remover, or no, nail polish remover. And if we want to find the hybridization of this atom, this right here, okay, what we're gonna do is we're gonna count how many atoms it's bonded to. So let's see here, it's bounded to 1, 2, 3 atoms. It's not bounded to 4 atoms because of the other bond. The double bond doesn't care at this point. It's just only the atoms, not the bonds. So 1, 2, 3. Okay, 3 atoms. 3 atoms. And count how many lone pairs it has. Alright, so what it means is how many lone pairs the central carbon has. This one, this stupid thing. How many carbon, how many lone pair it has. So a lone pair is when the electrons are like just chilling and doesn't bond. And if carbon can have 4 bonds in total, it cannot have over 4. Not like 5 or 6 or 7 or shit. Yeah. So it has 1, 2, 3, 4. It has 4 bonds, and that means it doesn't have any lone pairs. So 0, basically 0 lone pairs. And then we add them together. Oops. Yeah. Add them together. It's 3. And now look at here, look at this. We can see that if the sum is 2, it's sp. If the sum is 3, it's sp2 hybridized. If the sum is 4, then it's sp3 hybridized. We got 3. That means that this carbon atom right here is a sp2 hybridized carbon atom. And that's basically it. We can also do it to this methyl group. Like, let me show you. CH3. Let me draw the hydrogen since it's important. Alright, let's do this one then. Okay, we're gonna count the atom, and I want to find the hybridization of this carbon atom, okay? So 1, 2, 3, 4. Alright, we have 4. Now we're gonna count how many lone pair this MF have. 1, 2, 3, 4. It's occupied all its orbital. That means it doesn't have any lone pairs. So 0. It's 4. And since we got a sum of 4, that means that this carbon atom right here is sp3 hybridized orbital, or hybridized carbon atom. And yeah, that's basically it. That's how you can calculate the hybridization of atoms. So pretty easy when you stumble upon those questions in the exam. But yeah, I need to like fill this up, make it more pleasing to look at. But yeah. Oh shit! I already have an exam of the same damn okay. Yeah, okay. So hopefully it makes sense. Now we got hybridization patterns. So an sp3 can either look like this, so like this, or this. sp2 is always with double bonds, except when you see a cation like this, like a positive formal charge or carbon, then it's a sp2 hybridized molecule. If it's and a triple bond or like this is a sp hybridized orbital. Yeah. Okay, let's just do this just for fun. This was for my video, but we can just do it quick. Okay, so identify the hybridization state for the nitrogen atom is in ammonia. Alright, we have ammonia right here. This ammonia. Now we're gonna do our thing. So we're gonna count how many atoms this nitrogen has. Okay, so we're gonna say 1, 2, 3. It has 3 hydrogen atoms, or like 3 atoms it's bonded to. Alrighty, and then we have now we have to count how many lone pairs the nitrogen has. So nitrogen is in column 5A of the periodic table. That means it has 5 valence electrons. Okay, so if it has 5 valence electrons, then it doesn't have any like lone pairs. If it has less than 5, then it has lone pairs. Okay, so we're gonna count. So we're gonna count 1, 2, 3. Okay. That means the nitrogen needs 2 lone pairs. Alright, 2 lone pairs to fill up the 5. So 1, 2, 3, 4, 5, 2. So it's gonna look like this. And how many lone pairs is it? 2. So 5. Then that must be a SP. Is it 1 lone pair? No. Cannot be a lone pair. Then it must be SP3 at this point. Yeah, then it's SP3 hybridized. Okay. Yeah. Oh yeah. Yeah, then we have another like practice problem for the video. Probably it's gonna be good, so they understand. Also, this is a hardcore molecule. So for each carbon atom in the following molecule, so like this, 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 and this. This is each of carbon atom. And we have to determine their hybridization state. And then we have some Vesper theory. And what sp3 is, tetrahedral, trigonal planar, and liner, like I said in the beginning of the stream. Yeah, then a little speed run to like summarize it all. And what happens when one of those hybrid orbitals holds a lone pair? Then it gets like bent in shapes and shit. Yeah, so yeah, basically what is it? So yeah, ammonia. Wait, what? Ammonia has... Alright. Then we have water. Oh, okay, I understand. 1, 2, 3, 4, 5. Okay, I understand. It makes sense. It makes sense. The only 6 shapes you need. So when you see an sp3 with 0 lone pairs, it's a tetrahedral shape. Yeah, sp3 with 1 lone pair is trigonal pyramidal, and sp3 with 2 lone pairs is bent. And sp2 with 0 is trigonal planar, and 2 with bent. Yeah, all that. How to do this shit fast? Find the hybridization, so count the electron regions, what is also called, and count the lone pairs and pick the shape from the list. So yeah. And lone pairs, when lone pairs start acting different. Yeah, so lone pair not involved in resonance. sits in hyperorbital. So geometry follow normal Vesper rules. What? Yeah, and that's basically it. That's basically the video. So what I'm gonna do is I am going to wait to do this because I actually have something to read first. Like I have to study first. Alright? Oh shit. Alright. Let's see here. There we go. Problem C best, right? Should probably make it more smaller so you can view what's going on. Yeah, that's much better, right? Alright. Give me a moment. I'll be right back. Just need to test if everything works well. Just gonna check. Yeah, okay. The audio is good. Everything plays very well now. Honestly, OBS is pretty hardcore. Like, I don't want to jinx anything, but none of my frames has dropped yet. Anyway, let's read some. Alright, so substrate binding active site. The interaction that binds substrate to the active sites of enzymes include ionic bonds, hydrogen bonds, dipole, and wait, let me do something real quick. Like real quick. Let me see here. Does this shit work actually? No. Oh my God, have mercy. Wait, see this then? Oh, maybe onto something. Okay. Okay, interesting. There we go. This should be much better, right? Well, let me transition to it. There we go. Okay. This should be good, right? Yeah, okay. Alright. Let me... There we go. Alright. Substrate binding an active site. The interaction that binds substrate to the active sites of enzymes include ionic bonds, hydrogen bonds, dipole-dipole bonds, and ion-dipole interactions, as well as van der Waals and hydrophobic interactions. These bindings interactions are the same bonding interactions responsible for the tertiary structure of proteins, but their relative importance differs. Ionic bonding plays a relatively minor role in protein tertiary structure compared to with hydrogen bonding or van der Waals interactions, but can play a crucial role in binding of a substrate to an active site. As intermolecular bonding forces are involved in substrate binding, it is possible to look at the structure of the substrate and postulate the probable interaction that it will have with its active site. As an example, consider pyruvic acid, the substrate for lactate dehydrogenase. Okay, so we see that this side, the red side, is ionic, and the methyl group is van der Waals interaction, and the hydrogen bond is the O. Alright. Okay, it kind of makes sense. So the O right here bonds to the H, and the O and the methyl group does the van der Waals, and the negative charge does with the positive charge, which is an ionic bond, like sodium and chlorine, which creates table salt, and it's an ionic bond too. So that makes pretty much sense. Yeah, okay. If we look at the structure of pyruvic acid, we can propose three possible interactions by which it might bind to its active site. An ionic interaction involving the ionized carboxylate group, a hydrogen bond involving a ketonic oxygen, and a van der Waals interaction involving the methyl group. If these postulates are correct, it means that within the active site there must be binding regions containing suitable amino acid residues that can take part in these intermolecular interactions. Lysine, serine, and phenylalanine residues, respectively, will fill the bill. A knowledge of how a substrate binds to an active site is invaluable in designing drugs that will target specific enzymes. The catalytic role of enzymes. This section considers the blah 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 blah. Okay, nobody cares. Binding interactions. In the past, it was thought that a substrate fitted its active site in a similar way to a key fitting a lock, Fisher lock. Both the enzyme and the substrate were seen as rigid structures, with the substrate, the key, fitting perfectly into the active site. Okay, so the active site is like a lock and the substrate is a key. Okay, so it's like an analogy for that. However, this scenario does not explain how some enzymes can catalyze a reaction on a range of different substrates. It implies instead of that an enzyme has an optimum substrate that fits it perfectly, whereas all other substrates fit less perfectly. This in turn will imply that the catalyzed reaction is only efficient for the optimum substrate. As this is not the case for many enzymes, the lock and key analogy must be invalid. Okay. Okay. It is now proposed that the substrate is now quite the ideal shape for the active site, and that it forces the active site to change shape when it enters a kind of molding process. This theory is known as Koshland theory of induced fit. Sounds like a bodybuilding split. Since the substrate induces the active site to take up the ideal shape to accommodate it. For example, a substrate such as pyruvic acid might interact with specific binding regions in the active site of lactate dehydrogenase via one hydrogen bond, one ionic bond, and one van der Waals interaction. However, if the fit is not perfect, the three bonding interactions are not ideal either. For example, the binding groups may be slightly too far away from the corresponding binding regions in the active site. In order to maximize the strength of these bonds, this enzyme changes shape, such as the amino acid residues involved in the binding move closer to the substrate. Interesting. So the enzyme has ability to shapeshift. This theory of induced fit helps to explain why some enzymes can catalyze reactions involving a wide range of substrates. Each substrate induces the active site into a shape that is ideal for it, and as long as the molding process does not distort the active site so much that the reaction mechanism proves impossible, the reaction can proceed. The range of substrate that can be accepted depends on the substrate being the correct size to fit the active site and having the correct binding groups in the correct relative positions. However, it is important to note that the substrate is not a passive spectator to the molding process going around it. As the enzyme changes shape to maximize bonding interactions, the same thing can happen to the substrate. It too may alter shape. Bond rotation may occur to fix the substrate in a particular conformation, and not necessarily the most stable one. Bonds may even be stretched and weakened. Consequently, this molding process designed to maximize binding interactions may force the substrate into the ideal conformation for the reaction to follow, and may also weaken the very bonds that have to be broken. I was also thinking, like, after my after this video I'm gonna make about orbitals and geometry, I really want to make a video based on functional groups, because I feel like a lot of people has a difficult to memorizing functional groups, but in reality it's pretty easy, because all you have to do is like think of them as characters, like gaming characters, like Mortal Kombat. If you watching it had probably played, hopefully, or Tekken 7 or Tekken, and just think of them like characters, like fighting characters. So like carboxylic acid queen or something like that. But yeah, I'll definitely make a video about that with practice exercises, so you will master that instantly. Alright. Once bound to an active site, the substrate is now held ready for the subsequent reaction. Binding has fixed the victim, the substrate, so that it cannot evade attack, and this same binding has weakened its defenses, the bonds, so the reaction is easier. So it requires a lower activation energy. And the lower, the better in chemistry. If you have a high activation energy in chemistry, then get the fuck out of here, bro. There is another point relating to substrate binding. The binding interaction with the active site must be sufficiently strong to hold the substrate for the subsequent reaction, but they cannot be too strong. If they were, the product might also bind strongly and fail apart to depart the active site. This will block the active site of the enzyme and prevent it from catalyzing other reactions. Therefore, a balance must exist. Finally, it is important to realize that the enzyme also binds the transition state. Alright, so the transition state is where you see a reaction and then you see an arrow. That's a transition state. Involved in enzyme catalyzed reactions. Indeed, the binding interactions involved are stronger than those binding the substrate, which means that the transition state is stabilized relatively more than the substrate. This results in a lower activation energy compared to a non-catalyzed reaction. Okay. Alright, now we have some acid-base catalysts. Acid-base catalyst is often provided by the amino acid histidine, which contains an imidazoles ring as part of its side chain. The imidazoles ring acts as a weak base, which means that it exists in equilibrium between the protonated and free base forms, allowing it to accept or donate protons during a reaction mechanism. This is important as there are often very few water molecules present in the active site to carry out this role. Histidine is not the only amino acid residue that can provide acid-base catalysis. For example, a glutamic acid residue acts as a proton source in the reaction mechanism of enzyme HMG. Okay, what the frick is this, bro? Alright, case study 1. While aspartic acid and aspartate residues act as a proton donor and proton acceptors, respectively, in the other enzyme catalyzed reactions, and it's just chapter. And finally, tyrosine acts as a proton source in the mechanism by which the enzyme 17-beta hydrogen steroid type 1 catalyzes the conversion of estrone to estradiol. Alrighty, so the nitrogen has a lone pair. The lone pair attacks the H, and then after it attacks the H, the lone pair on the NH goes to that. Oh, so resonance. Okay, so resonance. So it goes there and then it hops there. Okay. Okay. Nucleophilic groups. The amino acids serine and cysteine are present in the active sites of some enzymes. These amino acids have nucleophilic groups in the side chains, OH and SH, so alcohol and ethyl, which are able to participate in the reaction mechanism. They do this by reacting with the substrate to form intermediates that will not be formed in uncatalyzed reactions. These intermediates offer an alternative reaction pathway that may avoid a high energy transition state and hence increase the rate of reaction. Normally, an alcoholic OH group, such as the one on serine, is not a good nucleophile. However, there is usually a histidine residue close by to catalyze the reaction. For example, the mechanism by which simotrypsin hydrolyzes peptide bonds involves in catalytic traits of amino acid, serine, histidine, and aspartic acids. Okay. Serine and histidine participate in a mechanism as a nucleophile and acid-base catalyst, respectively, while the aspartate groups interact with the histidine ring and serve to activate and orient it correctly for the mechanism. Okay, So that's a hydrolysis. Okay, so alright. The presence of nucleoserine residues means the water is not required in the initial stages of the mechanism. This is important because water is a poor This is important because water is a poor nucleophile and may also find it difficult to penetrate to penetrate the occupied active site. Alright. Secondly, a water molecule will have to drift into the active site and search out the carboxyl group before it can attack it. This will be something similar to a game of blind man's bluff. The enzyme, on the other hand, can provide a serine OH group positioned in exactly right spot to react with the substrate. Therefore, the nucleophile has no need to search for its substrate. The substrate is delivered directly to them. So it's like DoorDash. Water is eventually required to hydrolyze the acyl group attached to the serine residue. However, this is a much easier step than a hydrolysis of a peptide link, as esters are more reactive than amides. Furthermore, the hydrolysis of a peptide link means that one half of a peptide can drift away from the active site and leave room for water molecule to enter. A similar enzymatic mechanism is involved in the action of enzyme. I'm not gonna acetylcholinesterase. Okay, that's an enzyme because it ends with ase. And also the next one, pancreatic lipase and viral protease enzyme carried by hepatitis C virus. The amino acid lysine has a primary amine group on the side chain that will make it better nucleophilic group than serine or cysteine. However, the group is generally protonated and physiological pH, which precludes its acting as a nucleophile. Having said that, some enzymes have a lysine residue located in the hydrophobic pocket, which means that it's not protonated and can indeed act as a nucleophilic group. Alright. Stabilization of the transition state. As stated earlier, binding interactions serve to stabilize the transition state of the enzyme catalyzed reaction and thus lower the activation energy for the reaction. One example of this involves what is known as the oxyanion hole, which is present in enzymes such as bro, what is this tongue twister's piece of shit, bro? Such as yeah. This region is occupied by the oxyanion intermediate that is formed during the reaction mechanism described in figure. Okay. In the vicinity of oxyanion hole, there are peptide groups that form hydrogen bonds with oxyanion and help to stabilize its negative charge. These hydrogen bonds are also possible with the carbonyl group of the substrate, but the interaction is stronger with the oxyanion because of the negative charge. A negative charge oxygen atom is stronger hydrogen bond accepted than a neutral oxygen atom. Hydrogen bonding also stabilizes the development negative charge into transition state, leading to oxyanion. This results in a lower activation energy for the stage of reaction mechanism. Okay. So when an oxygen has a negative charge, it is much stronger than a hydrogen bond accepted than a neutral oxygen. Got it. Cofactors. Many enzymes require additional non-protein substances called cofactors. for the reaction that should take place. Deficiency of cofactors can arise from poor diet and leads to loss of enzyme activity and subsequent disease. Example, scurvy. Cofactors are either metal ions, example zinc, or a small organic molecule called coenzymes, example NAD+, pyridoxal phosphate. Most coenzymes are bound by ionic bonds and other non-covalent bonding interactions, but some are bound covalently and called prosthetic groups. Coenzymes are derived from water-soluble vitamins and act as a body's chemical reagents. For example, lactate dehydrogenase requires the coenzyme nicotinamide adenine dinucleotide. Okay, so that's NAD+. In order to catalyze the dehydrogenation of lactic acid to pyruvic acid, NAD+ is bound to the active site along with lactic acid and acts as an oxidizing agent. So it basically oxidizes it. During the reaction, it is converted to its reduced form, NADH. Conversely, NADH can bind to the enzyme and act as a reducing agent when the enzyme catalyzes the reverse reaction. Alright. Oh my god. Let me test if the stream is going well. Mm-hmm. Okay, everything's going well. So NADP plus and NADPH are phosphorylated analogs of NAD plus and NADH respectively and carry out redox reactions by the same mechanism. NADPH is used almost exclusively for reductive biosynthesis, whereas NADH is used primarily for the generation of ATP. And ATP is what gives us cell energy, like the natural energy outcome. A knowledge of how the coenzyme binds to the active site allows the possibility of designing enzyme inhibitors that will fit the same region. See case blah blah blah. Okay. A large number of enzymes contain a metal ion cofactor or themed metalloenzyme. For example, the cytochrome P450 enzymes involved in drug metabolism contain an iron ion which can intercharge between two different oxidant states, iron 2 or iron 3. This allows cytochrome enzymes to catalyze oxidation and reduction reactions. Other examples are the kinase enzymes responsible for catalyzing phosphorylation reactions and which serve as important drug targets. These contain a magnesium ion which helps to bind the coenzyme ATP onto the active site such that it can act as a phosphorylating agent. Several protease enzymes responsible for catalyzing the hydrolysis of peptide bonds contains a zinc ion as a cofactor and are categorized as zinc metal proteases. Examples include matrix metalloproteinases and I what angiotensin angiotensin converting enzyme. The role of zinc ion in aiding catalysis can be illustrated in the reaction catalyzed by histone deacetylase, a reaction that removes the acetyl group from the side chain of the acetylated lysine residue. Zinc is present in the active site as zinc-2 and is held in place by free amino acid, which is ligands. The reaction mechanism is similar to the mechanism shown in and involves the formation of an oxyanion with an aid of histine and aspartate residues. However, in this case, there are no serine residue to act as a nucleophile. Instead, our water molecule acts as the nucleophile. One of the catalytic roles of zinc is to activate both the substrate and the water by accepting both molecules as ligands. This also helps to position the water correctly for the reaction. Nucleophilic addition to carbonyl groups take place with histine removing a proton from water as it reacts. An oxyanion intermediate is formed which interacts strongly with zinc. Zinc also stabilizes the developing negative charge on the transition state leading to the oxyanion intermediate, thus lowering the activation energy for that stage of the reaction mechanism. Alright. So zinc has a role. I mean, it's good for your hair and nails also. Naming and classification of enzymes. The name of an enzyme reflects the type of reaction it catalyzes and has a suffix "ase." That's what I said. Indicate that it's an enzyme. For example, an oxidase enzyme catalyzes an oxidation reaction. It is important to appreciate the enzyme that can catalyze the forward and back reaction of the equilibrium reaction. This means that an oxidase enzyme can catalyze reduction as well as oxidations. The reaction catalyzed depends on the nature of the substrate, that is, whether it is reduced or oxidized form. Enzymes are classified according to the general class of reaction they catalyze are coded with EC numbers. Okay, so we have, So there are different classes: hydrolases, lysases, isomerases, ligases, transferases, oxidases. All right. Genetic polymorphism and enzymes. There are often subtle differences in the structure and protein of an enzyme between different individuals. This is due to the fact that DNA that codes for proteins is not identical form to person to person. On average, there is a difference of one base pair in every 1,000 between individuals, and this is known as a genetic polymorphism. Since the nucleic acid bases act as a code for amino acids in proteins, a difference at this level may result in a different amino acid being introduced into the protein. Often this has no observable effect on protein function, but not always. Some polymorphism can adversely affect the proper functioning of an enzyme and lead to genetic disease. Others can have an influence on drug therapy. For example, individuals differ in their ability to metabolize drugs as a result of this phenomenon. Polymorphism can also alter the sensitivity of an enzyme towards a drug, making it later less effective. This is a particular problem in anti-cancer, antibacterial, and antiviral therapies where drug resistance can develop through the survival of cells containing less sensitive enzymes. Okay. Regulation of enzymes. Virtually all enzymes are controlled by agents, which can either enhance or inhibit catalytic activity. Such control reflects to local conditions within the cell. For example, the enzyme phosphorylase A alpha catalyzes the breakdown of glycogen. 2-glucose 1-phosphate. Subunits. It is stimulated by adenosine 5-monophosphate (AMP) and inhibited by glucose 1-phosphate. Thus, rising levels of the product glucose 1-phosphate act as a self-regulating brake on the enzyme. Interesting. But how does this control take place? The answer is that many enzymes have an allosteric binding site, which is quite separate from the active site. This is where the agents control. Hello? This is where the agent controlling the activity of the enzyme bind. Where this occurs, an induced fit takes place, which alters not only the allosteric binding site but also the active site. Let me check if my stream is going well. Yeah, whatever, man. When this occurs, an induced fit takes place, which alters not only the allosteric binding site but also the active site. Agents that inhibit the enzyme produce an induced fit that makes the active site unrecognizable to the substrate. Okay. So the allosteric inhibitor makes the active site more thin so the substrate can't fit. So it just wanders away. Interesting. We might wonder why an agent inhibited the enzyme has to bind to a separate allosteric binding site and not to an active site itself. After all, if the agent could bind to the active site, it will directly block the natural substrate from entering. There are two explanations for this. Firstly, many enzymes that are under allosteric control are at the start of biosynthetic pathway. A biosynthetic pathway involves a series of enzymes all working efficiently to produce a final product. Eventually, the cell will have enough of the required material and will need to stop production. The most common control mechanism is known as feedback control, where the final product controls its own synthesis by inhibiting the first enzyme in the biochemical pathway. When there are low levels of the final product in cell, the first enzyme in the pathway is not inhibited and works normally. As the levels of final product increase, more and more of the enzyme is blocked and the rate of synthesis drops off in a graded fashion. Crucially, the final product has undergone many transformations from the original starting material, so it is no longer recognized by the active site of the first enzyme. A separate allosteric binding site is therefore needed that recognize the final product. The biosynthesis of noradrenaline in section blah 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 is an example of biosynthesis pattern. Okay. Secondly, binding of the final product to the active site will not be a very efficient method of feedback control, as the product will have to compete with the enzyme substrate if the levels of latter increases when the inhibitor will displace and feedback control will fall. Many enzymes can also be regulated externally. Looks at this in more detail, but in essence, cells receive chemical messages from their environment that trigger a cascade of signals within the cell. These, in turn, ultimately activate the set of enzymes known as protein kinases. The protein kinases plays a important part in controlling enzyme activity within the cell of phosphorylating amino acids such as serine, threonine, and tyrosine in targeting enzymes. Equivalent modification. For example, the hormone adrenaline is an external messenger that triggers a signaling sequence resulting in an activation of a protein kinase enzyme. Once activated, the protein kinase phosphorylates an inactive enzyme called phosphorylase B. This enzyme now becomes active and is called phosphorylase A. It catalyzes the breakdown of glycogen and remains active until it is dephosphorylated back to phosphorylase B. Alright. In this case, phosphorylation of the target enzyme leads to activation. Other enzyme may be deactivated by phosphorylation. For example, glycogen synthase, the enzyme that catalyzes the synthesis of glycogen from glycose-1 phosphate, is inactivated by phosphorylation and activated by dephosphorylation. The latter is affected by hormone insulin, and which triggers a different signaling cascade of the adrenaline. Protein-protein interactions can also play a role in regulation of enzyme activity. For example, signal proteins in the cell membrane are responsible for regulating the activity of membrane-bound enzymes. Alright. Isozymes. Enzymes having a quaternary structure are made up of a number of polypeptide subunits, and it's a combination of these subunits can differ in different tissues. Such as variations are called isozymes. For example, there are 5 different isomers of mammalian lactate dehydrogenase (LDH), a tetrameric enzyme made up of 4 polypeptide subunits. There are 2 different types of subunits involved, which are labeled H and M. The former predominates in the LDH present in the heart muscle, while the latter predominates in the LDL present in the skeletal muscle. Since there are 2 different types of subunits, 5 different isoenzymes are possible. HHHL or like a microwave. Isozymes differ in their properties. Alright. For example, the M4 isozyme, just gonna call it isozyme, in skeletal muscle catalyzes the conversion of pyruvic acid to lactic acid, and is twice as active as the H4 isozyme in the heart muscle. The H4 isozyme catalyzes the reverse reaction and is inhibited by excess pyruvic acid, whereas the M4 isomer sounds so Japanese. Honestly, it's not. Isomer. Alright, key points. Enzymes are proteins that act as the body's catalyst by binding substrate and participating in reaction mechanism. The active site of an enzyme is usually a hollow or a cleft in the protein. There are important amino acids present in the active site that either bind substrate or participate in reaction mechanism. So they're just chilling there basically. Binding a substrate to an active site involves intermolecular bonds. Substrate binding involves an induced fit where the shape of the active site alters to maximize binding interactions. The binding process also orientates the substrate correctly and may weaken crucial bonds in the substrate to facilitate the reaction mechanism. The amino acid histidine is often present in active site and acts as an acid-base catalyst. Glutamic acid, aspartic acid, and tyrosine also act as acid-base catalysts in some enzymes. The amino acids serine and cysteine act as nucleophilic in the reaction mechanism of some enzymes. In some enzymes, lysine can act as a nucleophile. Alright. Cofactors are methyl ions or small organic molecules that are required by many enzymes. Coenzymes can be viewed as the body chemical reagents. Prosthetic groups are coenzymes that are bound covalently to an enzyme. Enzymes are regulated by internal and or external control. External control involves regulation initiated by a chemical messenger from outside the cell, and which ultimately involves the phosphorylation of enzyme. Allosteric inhibitors bind to different binding sites from the active site and alter the shape of enzyme such that the active site is no longer recognizable. Allosteric inhibitors are often involved in the feedback control of biosynthetic pathways. Isomers, not this again. Isomers are variation of the same enzyme. They catalyze the same reaction but differ in the primary structure, substrate, specificity, and tissue distribution. The amino acid sequence in enzyme may differ between individuals due to genetic polymorphism. This may or may not result in difference in enzyme activity. Alright, next chapter is about enzyme kinetics. Interesting. Okay, so that's a very short what's next? Receptors. Interesting. Alright. Gotta read that tomorrow, next stream, honestly. Alright. Now. Yeah. So now I'm gonna make my video done. Let me see here. Or if you want me to read more, you can just tell me in chat. Chatter, my chatter. What is a covalent bond, huh? Okay, so why the hell is my cast not working? Was? What the hell happened? Wait. Oh, there we go. Just need to restart my shit. There we go. Can you see this? Yeah. Okay. Oh my god, the whole shit just crashed. Oh. Yo, what? Alright, now I'm gonna just fill this shit out. Hopefully you're all doing well. Those who's watching. Currently have 4 viewers. I don't know if that's myself 4 times. Probably is. No, actually I have 0 viewers. My bad, G. Yeah. Whatever, man. I'm not doing this shit for viewers. I'm just doing this for myself. Doesn't matter. It will be worth in the end. Okay. This is the Y axis, and this is the Z axis, and this is the X. But now I have to draw a shape. Copy that. What? Up. Mhm. Yeah, 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 yeah. Not so. think I'm gonna end the stream this time. Yeah. Also gonna record some shorts for this video as well. So practice problems. And yeah, the grind is definitely on. I'm not gonna stop anytime soon. So expect the best of this channel, and hopefully you will enjoy every single bit, and hopefully you will ace all the exams. But yeah, that's all. Thank you for watching, if you did actually watch and just chilled, maybe while doing homework, I don't know, but yeah. My throat is dying, but eat well, stay hydrated.